For today, we're going to be discussing antimicrobial chemotherapy. We talked extensively last time about microbial control and the differences between disinfecting inanimate objects versus living organisms. A lot of the chemical agents that we discuss that are able to be used to sterilize or disinfect inanimate objects are actually too potent to be used internally and can have a lot of toxic and negative side effects if they're used in that manner. So instead, in order to treat infections that affect living organisms, we can use alternative methods like chemotherapy. This is the use of chemical agents to treat disease, but instead, we are choosing drugs that are implemented to do things that don't interfere with our normal cellular function, but rather only target the pathogens instead. The way that these types of chemicals can affect pathogens is by interfering with their nutrition and growth or the integrity of the cells themselves. So anything that would be considered normal cellular activity, like metabolism, cell division, cell wall formation, any of those at all, if you can obstruct them at any one of the many steps that are required, will do the trick. Microbial control is one of our best tools. Chemotherapeutic agents can be used to both fight and treat diseases or prevent them from coming on at all. Most of them are antibiotics. There's a difference between things that are synthetically manufactured and what antibiotics are. Antibiotics are different materials that are used by cells to affect other cells that we can then study and then produce artificially. But their foundation is actually from cell versus cell warfare. A lot of what we know about killing bacteria actually comes from studying bacteria trying to kill others that are in their environments, competing for space or nutrients, and that have implemented their own chemotherapeutic strategies to get rid of their competitors directly. And those are our antibiotics. There are other drugs that we have devised synthetically that could be derivatives of antibiotics or have a similar structure or function to them, but those are not considered antibiotics since they are man-made and they're not made in nature. And just to stretch way back into this course, the first person that ever discovered an antibiotic was Alexander Fleming, and it was penicillin. He was growing some bacteria up in culture and had his plates get contaminated with molds. The molds that were growing actually started to secrete an agent that created that zone of inhibition where bacteria couldn't grow because that chemical was present in relatively high concentrations. So this was the first observation of microbe on microbe warfare. And after the contents of that agar was isolated, penicillin was discovered and was then used and implemented in treating human diseases not too long later. And while a lot of scientific discoveries happen because we're obviously looking into things and trying to figure out how things work, some conclusions come completely by surprise, like this one. So after penicillin was discovered, it kind of set the stage to look into, well, what else exists that can also exhibit this type of functionality? And there were more compounds that were produced by one microbe to kill off another that were slowly discovered and characterized. And this has actually created the repertoire of medications that we still currently employ and use in medicine in this current day. There are a lot of considerations that have to go into a chemotherapeutic agent, and there are desirable characteristics that have to be determined to see whether or not something is going to be a useful one, because you can have a lot of drugs that will kill bacteria, but are they going to harm the host? That's one of the big questions. Anytime that you're administering a therapy, you have to always consider whether the risk of the therapy is worth the treatment. And in the cases of chemotherapeutics, you're looking to see if side effects that are caused by these medications are worth the effect of killing the affecting microbe. One really important aspect is that selective toxicity. The drug has to kill or inhibit the pathogen while impacting the host as little as possible. The selective toxicity can be expressed in a couple of different ways. One of the things that we have to determine is what is an appropriate therapeutic dose. This is the level that's required for clinical treatment of a specific infection. If you exceed it, yeah, it can still get the job done, but maybe there are some adverse side effects that come along with that. If you administer too little, it might not get rid of the infection completely. So there is a delicate balance that needs to be found. There's also that toxic dose, which is a level of the drug where it would become too toxic for the host. What we get from those two variables is called the therapeutic index, and that's expressed as a ratio of those two factors. The larger the index, the better the drug. And so if we take a look at the equation down below, toxic dose over the therapeutic dose equals the therapeutic index. Another desirable trait is that the drug would not necessarily impact cells of the host. So if you have a microorganism that you're trying to target, you would assume that there would be certain characteristics, whether they're physical or chemical, that those entities would have that are not common to the host. So thinking about all the things that we know about microorganisms and how they're formed, you can probably think of a short list of things that prokaryotic cells have that eukaryotic cells do not. One great example that comes to mind is peptidoglycan. That is a big target that we actually exploit. There are differences in ribosomes. There are differences in the types of amino acids that are present in bacteria versus eukaryotic cells. There's a huge number of these differences that can be characterized and targeted. So that's what we start looking for when we're trying to develop new drugs. After you figure out what might be appropriate in that fashion, the next step is to make sure that that particular drug can't be inactivated by a pathogen. Just because it can target them, it doesn't mean that that pathogen won't have some mechanism to circumvent your efforts. Lots of organisms will actually have enzymes that can cleave up medication so that they're no longer effective, or if they happen to get into the cell, a lot of cells have efflux pumps to send that right back on out of the cell membrane into the extracellular environment. Getting your drug to its target and retaining it there are two really important aspects of this entire process. The other part to consider is how long will the drug stay within a person? Over time, if you have multiple doses over several courses, is that going to accumulate in a system? And if so, can it accumulate to toxic levels? How is it going to get processed by the body? Is it going to tax our organs that are responsible for that type of work, like the liver or the kidneys? There's lots of things that have to be considered. Depending on how medication is administered can also affect its retention time. You can see here if penicillin is introduced into the body in a bunch of different ways, it'll stay in the body longer or shorter. In the case of an injection, where it's entering the body intramuscularly, so into muscle tissue directly, that's going to have a lot shorter of a time frame inside the human body because it's going to get put directly into the bloodstream. You're going to have vascularization in those muscles. That material is going to get picked up and spread around the body a lot more quickly, which means it's going to get processed a lot more quickly. If we look at the oral route for penicillin G, that's going to be slower because there is the administration orally and then the absorption process. It'll still get into the bloodstream, but it'll be a lot slower. So depending on how you want these things to be administered and the length of time, remember that length of killing that we talked about, how long something has to be in contact with a chemotherapeutic agent to be effective, are things to think about when we're looking at the administration route. There's also the spectrum to consider because when we think about prokaryotic cells, a lot of cells will share similar features. Like most bacteria, for example, will have peptidoglycan in their cell walls, while only a few of them might have mycolic acid in their cell walls. So if you're targeting one of these two things, you're looking at a spectrum. How many microbes will your drug target? If you're looking at narrow spectrum drugs, these are going to be specific against a small number of pathogens, whereas a broad spectrum drug is going to attack a lot more of them. And that's simply based, again, on how ubiquitous a target that you're trying to exploit is found in many different organisms. So this chart down here is just giving a couple of different examples of some common antibiotics and other medications and how broad or narrow their spectrum is. So if we look at something like tobramycin, for example, this is only going to target a small portion of mycobacteria and gram-negative bacteria. Penicillins will deal with gram-negative, gram-positive, and chlamydias, whereas tetracyclines will deal with most bacteria, with the exception of those that contain that mycolic acid in their cell walls. So hopefully this will give you a good picture for what we've just been discussing here. There's also the idea of antimicrobial effectiveness. So whether or not drugs are going to kill microbes or just simply inhibit their growth. And again, the difference in suffix is cidal versus static. These traits can vary depending on the species that's being treated and the concentration of the drugs administered or the dosaging. So there's two different values here that can be ascertained based on species-specific treatment with drugs. There's the minimal inhibitory concentration, which is the lowest level of a drug at which it will prevent the growth of a specific pathogen, so inhibiting growth, or the minimal lethal concentration, which is the lowest level that will kill the pathogen. So what is the minimum amount of a product that's required to kill something or inhibit it? And based on all of the numbers for all of the different medications that would target a specific species, you can decide on which medication would be best to treat a specific kind of infection. There's also the potential of combination therapies, so you can offer drugs alone or in tandem with others. There is synergistic effects from time to time where the effects of two drugs together is greater than the effect of either one alone. You can see that in this example here, where we've got two different kinds of antibiotics that have been applied with the disc diffusion method that we touched on previously. And you can see that on their own, they do both inhibit bacterial growth. But in between where the two meet up, there is a much larger effect, the synergistic effect, where both of those drugs together cause more inhibition. So it would be beneficial if you're trying to treat something to add those two things together in order to have a larger effect without having to increase the dosage to a potentially higher and detrimental level for the host. There's also the possibility of the opposite occurring as well. Antagonism can occur when two drugs together are less effective than either one of them are alone. So imagine the opposite effect here, where we've got two drugs side by side, each having their own individual effect on the same species, but when you put them together, they may interfere with one another or interfere with their efficacy so that the bacteria would not be inhibited as much. So when we're testing these things in the clinical setting before opening them up to the general public, there are three major factors that have to be met. The drug has to be able to reach the site of an infection. So if you have an infection, how are you going to administer it best to get there and to be effective at the place where it needs to be? If it's oral administration, you have to make sure that your drugs are non-toxic if they're going to get absorbed into the bloodstream, and that they're also relatively stable. You can't have them degrade too soon. There's also the option of parenteral administration, which would be below the skin, where because they're being taken up right away into the bloodstream, they can be a little bit more toxic or potent in nature, and they can be broken down faster by the host versus needing to be more stable for that absorption period. Additionally, the pathogen obviously has to be susceptible to the drug. You're not going to treat a cell with a type of medication that wouldn't be effective for them in their specific types of processes that they might have. The agent used also has to exceed the pathogen's MIC value to be effective as well. There's a variety of different laboratory tests that can be employed to evaluate the effectiveness of a drug, and these things include the dilution susceptibility test, the disc diffusion test, and the E-test. The dilution susceptibility test is used to determine the minimum inhibitory and the minimum lethal concentration values. So what we do is we add serial dilutions of antibiotics to broth. And each broth tube is going to contain a little bit less antibiotic than the previous one, if you've forgotten what a serial dilution looks like. Once all of the dilutions are prepared, you would add the same amount of pathogen to each tube, and then the growth is measured. And again, we measure growth by things like turbidity. The minimum inhibitory concentration from all of the different concentrations tested is the lowest one of the antibiotic that will result in no microbial growth after 16 to 20 hours. And the minimum lethal concentration is determined by taking the different tubes that have been inhibited and putting them into media that is lacking the drug and seeing if anything grows. So you take these cells that might just be inhibited and put them into new growth media. If those cells start growing again, obviously they're not dead, but if they don't grow, then you know that that drug was effective at killing them. And from those, you look at the concentration that was the lowest where cells were killed. So this is a great example of some wells that have been inoculated with bacteria with varying concentrations of drugs. So here you can see we're testing a certain bacterial species against a bunch of different medicines. Up here you can see doxycycline, for example. We have growth in all of the wells of this experiment, meaning that none of the concentrations of doxycycline that were applied were effective in either inhibiting or killing that particular species. For sulfamethoxazole, you'll see that we do have inhibition in some of these wells, but not completely. And as that concentration decreases, we do still see growth, but there are concentrations here where a lot of the growth is being inhibited. Streptomycin, no matter what the concentration here, is extremely effective because you see that there is no cell growth in any of these. Whereas in this final example, you can see ethambutol and kanamycin are equally effective. So they have a similar concentration where they are effective at inhibiting or killing this particular species. So just at a quick glance, this tells us a lot about this particular species and the medications that would be effective to treat it well. For the disc diffusion method, we've already touched on this a little bit, but what we essentially do for this is grow up a broth culture and then we make a spread plate. So we're looking for that bacterial lawn, and we're going to see this in the laboratory as well. What we do then is we spread it around on the plate until it's evenly distributed, and then it's all going to grow up nice and uniformly. You add discs that contain different types of antibiotics, and you can see on this plate there's a bunch of discs that have different codes. All of these are going to correspond to different medications, perhaps at different concentrations, but mostly you test different types of medications in this method. So when the discs are applied to the plate, the medication diffuses outward into the agar, which will create a concentration gradient away from the point where it touches. The most concentrated will be closest to the disc, and as it spreads out, it'll become less and less concentrated. Following an incubation period, you can actually measure the diameter of that zone of inhibition and determine the minimum inhibitory concentration. The larger the diameter that you get, the smaller the minimal inhibitory concentration will be. So you can see in this example here, this particular drug was the most effective at inhibiting growth because that zone of inhibition is the largest. Some drugs on this plate were not effective at all in inhibiting growth. So relative to one another, you can determine which type of medicine is going to be more effective compared to the others based on the size of that zone of inhibition. The E-test is similar to the disc diffusion method, except it's a little bit more quantitative in nature. You have a semi-quantitative analysis with the disc diffusion method, but the E-test is going to give you a really great numeric value. In a similar fashion, you prepare a bacterial lawn, and then what you do instead of applying discs is you apply a test strip. The strips come pre-made with a gradient of a specific antibiotic drug, and you place those onto the agar surface. And then you can see right on the strip where the minimum inhibitory concentration is based on that lowest concentration where you see bacteria not growing. So you essentially look at where the edge of that zone of inhibition is, and that's going to be your minimum inhibitory concentration. Depending on the drug, you'll see that different ones will have a different value because they'll be more or less effective depending on what they are and what they target. There are many types of classes of drugs, and they're categorized based on what kinds of targets they exploit. The mechanism of action can be inhibitors of cell wall synthesis, protein synthesis inhibitors, metabolic antagonists, nucleic acid synthesis inhibitors, or they may directly damage cell membranes and kill cells that way. And you can see here that this schematic just kind of shows some of those ideas where you can see up here, if we inhibit the cell wall formation, then obviously cells are not going to have their integrity. If we inhibit the synthesis of proteins, lots of those enzymes that are required for cellular work will not function. If we inhibit the replication and transcription of different genes in the DNA, cells can't divide well. If we injure the membrane again, that's similar to what we saw up here. We compromise the integrity of the cell, which can cause leakage. And of course, the inhibition of enzymatic activity can cause a lack of metabolites or other things that would necessarily be required for normal cellular function. So anytime you interfere with one or all of these, you're potentially limiting cells in terms of what they can do or killing them. To take this a little bit further now, there are lots of examples of drugs that do all of the different things that we just discussed. And I don't want you to necessarily memorize all of these, but we are going to take some time to discuss a couple of examples and what they specifically do in the coming slides. First, we're going to talk about synthetic metabolic antagonists. An antagonist, generally speaking, is a molecule that mimics another one that is normally used by a cell. And what it does is when it's present, it will compete with or obstruct the normal processing of that native molecule. let's go back to the thoughts around competitive inhibition of enzymes. That is an example of an antagonistic inhibition method where you're introducing something that's similar to that enzyme substrate, but that is not the substrate. So it occupies the enzyme's time and decreases, or if it binds well, can even inhibit that enzyme's activity altogether. When we look to treat cells with certain medications, typically we're going to target metabolism. Metabolic antagonists are synthesized in the lab setting, and they're going to be analogs to molecules that are normally needed for regular metabolic activity within the cell. So they're going to be similar to the molecules that would be used in metabolic pathways, but different enough that they're once again going to compete with the regular molecules to cause problems. We make analogs that can replace vitamins, amino acids, purines, or even pyrimidines, and this will greatly impact the ability of cells to do what they need to do. Sulfa drugs are a great example of this, and they're actually analogs of para-aminobenzoic acid, or PABA, and this is a precursor for folic acid, which is a material required by bacteria. So if you have a look down here, you'll see sulfonamides and PABA are very similar in structure. And typically, the pathway is that PABA gets incorporated into the formation of folic acid. So if we supply cells with a lot of this instead, they're going to be taking this up and not be able to make the folic acid that they would normally require. Unfortunately, there are a lot of species that are resistant to this type of treatment because they have found ways to work around this and will actually import external sources of folic acid. They say, you're going to prevent me from being able to make it? Well, fine, I'll just order takeout from the extracellular environment instead. There are some workarounds for this that involve a couple of different types of medications that will block folic acid production altogether. And this is done in combination therapy to prevent folic acid production and force any species that are resistant to these changes to have two specific mutations to get around it, which is very unlikely. Isoniazid is a growth factor that's an analog with very narrow spectrum activity because it only impacts cells from the Mycobacterium species. And what it does is it interferes with the production of mycolic acid. If these cells can't make that waxy layer that is impermeable to all different types of treatments, it is weak and susceptible. We also have a large class of drugs that can inhibit DNA function. Some synthetic drugs can get into DNA and actually inhibit the replication prior to cell division or transcription of genes as well. The only problem with this is that they can also potentially interfere with the host DNA or RNA because we don't have a lot of differences between ours and prokaryotic genetic material. While there is a bit more risk involved with this type of therapy, some drugs are very highly specific, like rifampin and quinolones. Quinolones in particular will inhibit the actions of DNA gyrase, which you may remember is responsible for nicking and supercoiling of DNA prior to packaging within the cell. And don't forget that the sheer volume of DNA cannot be packaged into a cell if it is not supercoiled. So without DNA gyrase, these cells simply cease to exist because they cannot pack their DNA inside. There are also a variety of analogs that can be implemented where we're taking either amino acids or nucleic acids and replacing them with analogs. And you'll see here that structurally these molecules are very similar, but they have one atom that has been added to them in a lot of cases that makes them different enough to alter their chemical properties. So they will not function correctly when they get incorporated into molecules as they're being built. So imagine proteins or DNA or RNA are not going to have those chemical properties that they would need to be correct. So we'll move on now to discuss some antibiotics. And again, these are things that occur naturally and are used by different microorganisms to kill others that are in their environments or competing with them. We have identified a lot of antibiotics, but unfortunately, a lot of them aren't particularly useful or potent enough to be used in human hosts or mammalian hosts. A lot of the antibiotics that we do use are chemically modified in the lab in order to increase their efficacy. And because they're modified, they're considered to be semi-synthetic. Part of their alteration prevents them from potentially being inactivated by the target pathogen. And some examples of these are methicillin, ampicillin, and amoxicillin. There are a lot of bacteria that produce antibiotics naturally, and you can see here that Streptomyces makes a lot of different things. You don't want to mess with Streptomyces and get in on its turf because it'll probably try to kill you, as you can see here. Other species will still produce some, but we do see that a lot of the ones that will probably stand out, streptomycin, tetracycline, kanamycin, these are all ones that we're probably very familiar with. Bacitracin is another important one that we'll discuss. And of course, on the fungi side, penicillin is one that's quite popular. Again, this is not a table that you need to memorize, but rather just something to help to feed this discussion, to show you that there are a lot of diverse products out there. And not only that, ones that have yet to be discovered. There are many, many bacteria that exist out there that have not even been found yet in some of the most remote areas of the world where things are still being researched regularly. So maybe there's a whole slew of new antibiotics that we don't even know about yet. This is a small survey of some of them that are medically relevant to us. The inhibition of protein synthesis is good to target in prokaryotic cells because we do have slight differences in ribosomes. Unfortunately for us, though, some of our organelles also contain prokaryotically structured ribosomes as well. So we do have to do a lot of careful planning when we look at drugs that target these particular types of organisms. Eukaryotic cells have 80S ribosomes, while prokaryotic ones have 70S, which is really a differential in the size of one of their subunits. Many of the drugs that we apply will interact with either one of the two ribosomal subunits and cause them to not function properly. Depending on the drug in hand, you can affect different parts of the protein synthesis process. So are we looking at the initiation sequence? Are we looking at the elongation step? What part of the process are we trying to inhibit? And any step along the way is fair game. And even though you've got two different drugs that might affect the same step in a major process, they may function in completely different ways. So up here, we've got a little schematic of what a prokaryotic ribosome is going to look like here, with the 50S subunit on top and the 30S on the bottom, so the large and the small, of course. These are some examples of some drugs that are going to impact different parts of this process. Streptomycin is going to change the shape of the small portion of the ribosome, whereas tetracycline will interfere with the attachment of transfer RNAs to the mRNA ribosome complex, so it'll prevent this process from happening. No codon recognition. Erythromycin is going to bind to the 50S portion, so the large portion, and will prevent translocation, which will stop the ribosome from being able to move down the mRNA. Or even chloramphenicol, which will bind again to the 50S portion, but will inhibit the formation of the peptide bond. So we're not going to have the chain transferred well to the new amino acid. Any one of these medications is considered something that will inhibit the big picture of protein synthesis, but the mechanisms in which they do it are all different. So in terms of what I would expect you to know for this, what I would hope is that you will know the names of the drugs and what types of processes that they inhibit. So if I gave you a multiple choice question and asked you, which one of the following inhibits protein synthesis, you should be able to pick out tetracycline. When we get into the inhibition of cell wall synthesis, penicillin is the biggest one. It is going to prevent the synthesis of peptidoglycan because it interferes with those transpeptidation reactions. If you recall, we have NAG and NAM sugars that have amino acids that are conjugated to those, and to form those cross links that are going to attach all of those peptidoglycan polymers together to form an even larger complex, that transpeptidation reaction has to occur. Penicillin blocks the enzymes that do that cross-linking process. So the cell wall still forms without much issue, but it can't be put together. It would be like piling up a bunch of bricks, but without any mortar in between them. The wall is still there, but it's very weak. Other medications that do similar things are cephalosporins, which will bind to these penicillin-binding proteins to prevent peptidoglycan transpeptidation as well. And vancomycin will bind the amino acids in peptidoglycan to prevent the transpeptidation reactions from occurring. So you're either affecting the actual molecules that should be bonded together, or you're impacting the enzymes that would do that work to begin with. So you can see here in this diagram, we've got different types of medications that we just discussed here, showing their structure. You'll see that they all have this very unique form inside of them called the beta-lactam ring. And based on this diagram, you can see how they actually interfere with the formation of the cell wall. So here you see that beta-lactam drugs like penicillin or cephalosporin are going to interfere with the formation of those peptide side chains that we just talked about. The other hand here is vancomycin, which is actually going to prevent the amino acids from being able to bond to one another instead. And again, when we inhibit cell wall synthesis, we do leave cells susceptible to death. You can see in this example here, we've got some intact cells, whereas over here, the cell wall or cell membrane integrity has been compromised. Anytime you have leakage in a cell, it's lethal because all of those nice internalized factors like cytoplasm, ribosomes, DNA get exposed to the extracellular environment and start leaking out, which is not ideal and will lead to death. Injury to the plasma membrane is similar in its effectiveness because certain antibiotics can cause changes in the permeability of that plasma membrane. Similarly to eukaryotic plasma membranes that we've been talking about all along, having semi-permeability is extremely important. You want certain molecules to be able to pass through. You want to prevent most molecules from being able to pass through. And anytime you just start poking holes in that membrane, all of that goes out the window. Polymyxin B is a drug that's going to disrupt the plasma membrane by attaching to the phospholipids that make it up. So they're no longer able to associate with one another, but have these big drug pockets in between them that force them apart. As such, the permeability is going to be more, and things that should not necessarily be passing through can pass through. And this includes the cell's internal contents into the external environment. Daptomycin is another medication that can treat gram-positive cell infections, and what it actually does is depolarizes the cell membrane. You may recall that polarization of the cell membrane is important in transport and other types of metabolic mechanisms where we have to get molecules into and outside of the cells, and as soon as that depolarization reaction occurs, cells are not able to move molecules effectively. There are also a variety of antiviral drugs available, and 60% of all infections are caused by viruses. The problem with treating them is that they tend to be intracellular in nature, meaning that they go inside of our host cells. So it's difficult to target them when they're hiding inside of something that is considered to be part of us. In turn, most antiviral strategies end up targeting host cell structures. And as a result, there are very few antiviral drugs on the market. The ones that are out there can only really deal with a couple of specific viruses, and we're going to talk about a couple of examples of these. You can see when we're looking at the synthesis of normal viral DNA guanine nucleotides, we've got the nucleoside with the phosphate that's going to come in to form the nucleotide. That can get incorporated into DNA without too much issue. When we administer the analog, though, we have phosphate that's going to get bonded to a cyclovir, which is very similar to that nucleoside structure. And this is a false nucleotide that cannot be incorporated into DNA. As a result, the assembly of DNA is going to halt. Interferons are an intracellular method that our body uses as part of its immune response. If a cell gets infected by viruses, it actually can send a message out into its immediate environment to warn other cells that there's an infection in the neighborhood. So an infected cell is going to start to secrete all of these proteins called interferons into that environment to go and act on receptors of nearby neighbors. The neighbors, once they detect that there is a problem nearby, can start to produce antiviral proteins. These might be proteins to prevent the uptake of the virus. They might be enzymes to break it down once it gets into the cell. There's a variety of mechanisms that could be at play depending on the species, but interferons are a very effective intracellular method to prevent viral infections from spreading from cell to cell. Now, we've also talked a little bit about antibiotic resistance, but we're going to dive here into a couple of mechanisms that prokaryotic cells can employ to resist our best efforts to kill them. One thing that they can do is actually get rid of the target that the medicine is targeting. If it's a specific kind of protein in their cell wall or in their cell membrane, they can just stop producing that if it's not something that is essential to them being able to survive. They may be impermeable to antibiotics. Gram-positive bacteria have thicker cell walls and are more difficult to treat than gram-negative ones. They can also alter antibiotics to an inactive form. We just talked about how different antibacterial drugs have that beta-lactam ring. And some bacteria produce enzymes called beta-lactamases that come in and render those medications ineffective. They can also alter the target of the antibiotic. They can develop a resistant biochemical pathway, so an alternative mechanism to get around metabolic inhibition, or they can simply just pump drugs back out with efflux pumps. Antibiotic resistance is actually an enormous concern because as we are prescribing these things more and more, we're seeing more and more resistance cropping up all over the place. There are some regions of the world where antibiotic prescriptions are completely unregulated, and this is due to a lack of training and infrastructure. So some people are getting antibiotics from individuals that are not healthcare professionals. Some of them are actually sold to treat problems that are not related to infection at all, like headaches. If we look at the statistics, many of the antibiotics that get prescribed are completely unnecessary and only add to the problem of antibiotic resistance. And there is a lot of pressure. If you go to the doctor and you're unwell, you want to get better and you want to be able to get rid of your symptoms. The problem becomes when people pressure healthcare professionals to prescribe these things. Or there might be some doctors that just don't want to have to argue with somebody that wants some kind of treatment method and fast. So these things do get prescribed a lot more than they possibly should. We also have to consider that there's a huge use of antibiotics in the farming and the food industry. Lots of the animals that are bred for meat products are pumped full of antibiotics, and that's another mechanism that we are ingesting some of these things. Another huge problem is the underuse of antibiotics. If you are prescribed a course, let's say for an entire week, you should take that for that whole 7 days. Some people stop after day 3 when they start to feel better. And what that does is leaves some of those pathogens alive, which are resistant to that medication. If you do not take your full course, you're contributing to that resistance. Prescribing combination therapy can help with this, but really just making sure that you're taking your medicine as prescribed is a better method. So this is a diagram that just kind of summarizes the things that we've just been discussing, where we have overprescription of antibiotics in various formats here, and then also the use of antibiotics in animal feeds on farms. All of them come together to form that antibiotic selective pressure, where we end up eventually with more resistance to antibiotics as these different microorganisms are gearing up against them. We see a lot of that bacterial conjugation or that horizontal transfer of antibiotic resistance genes on plasmids. And this will produce strains that have a unique survival ability. Thankfully, there are a lot of groups out there that are still looking for new answers and new therapies, but this is a real problem that is becoming more and more prevalent as the years tick by. So this is a great summary about how antibiotic resistance genes can actually function. We can see here in the plasmid DNA, these are these non-essential genes that can be transferred back and forth between bacterial species through conjugation, and they can code for a variety of different things, like enzymes that can change the antibiotic, right? Or microbial products that can be produced to degrade the antibiotics so that they never reach the cell. We can have microbial products that deactivate the antibiotics, or form channels or pumps that can just send that stuff right back out from where it came from. And all of these mechanisms are protective for the bacteria that can implement them. Because a lot of these genes are carried on plasmids, there are many different ways, not only just conjugation, but several others, that can be used to transfer this resistance between different types of cells. We've talked about these before, so this is just a bit of a review here, but transformation, conjugation, and transduction are all mechanisms that are summarized here. Transformation is, again, when bacteria will pick up DNA that is in its immediate environment. It might be from a dead neighbor that has spewed out all of its cellular contents, and a cell might take up some of that DNA. Or conjugation, like we discussed, through the passage of a sex pilus between cells. And then finally, we have transduction, where bacteriophages that are infecting different bacterial cells will transfer bits and pieces of genes and DNA that can eventually get incorporated and expressed in a target. A couple of interesting facts for you. There were some freeze-dried E. coli from 1946 that contained plasmid genes for antibiotic resistance to streptomycin and tetracycline before they were widely used. In addition, other strains carried genes for resistance to semi-synthetic penicillins before they were ever synthesized. So these genetic adaptations are very widespread and are already established in a lot of different species, and it's only getting worse. As we already kind of touched on, scientists are starting to predict that determining and discovering new and effective alternative treatments is inevitable as these species become more and more resistant to our current interventions. There are a couple of different companies working to test new antibiotics, but they are very time consuming and costly to develop because many bacterial strains are becoming resistant to even the most potent drugs that currently exist. There is talk of returning to the pre-antibiotic age. And what would that mean for us? How would we persist on Earth if we no longer have effective treatments to be able to combat some of these pathogens? These are a lot of questions that we don't necessarily have the answers to. But thankfully, we're always on that expedition for new knowledge that can help to arm us against the things that are trying to wipe us off the face of the earth. One bright light at the end of this tunnel is that we can keep producing new analogs. We've already got the basic structure of a lot of drugs that work well, and we can test other analogs of them to see if they also have efficacy. Even slight changes in the molecular structure might be enough to be able to give hosts an edge in the fight with these types of treatments. For some context, approximately 7 million candidate compounds have to be screened every year to find a single useful drug. That's not to say that we won't find them, but we just really have to look hard to see them. And there is productivity from these methods. Something like saquinavir, which is a protease inhibitor, so inhibits protein-degrading enzymes, was discovered as a treatment for HIV. And this was done completely through computer-directed drug design, so a program that was meant to alter and change the structure of molecules and produce something that could be viable as a treatment. And we found something, and that's awesome. There are a lot of research groups that are looking how to select for new compounds from soil microbes. One of the biggest limitations is that a lot of organisms that exist out in the world cannot be easily grown in the laboratory setting. If we cannot grow them, we can't really study them very well. And as a result of that, we can have so many different bacteria that are producing these wonderful compounds that we just simply don't know about yet. Soil microbes are some of the most abundant in the world in terms of environmental density. And there is a really cool article here, if you want to watch, that shows some research groups that are working on culturing those bacteria in a way that they never have before in the laboratory setting. There's also the potential to lean on that bacteriophage therapy that we also discussed. And just as a bit of a reminder, these are therapies where we use viruses to target and destroy bacteria that we don't necessarily want around. The only problems with bacteriophage therapy is that there's a lot of work to be done to get them into a state where they could be used regularly. They're very promising, and the science is there to support that they would work well, but it's just a matter of, again, working through it enough to identify the risks and the rewards and to refine them enough to be used in the day-to-day. So hopefully this discussion got some of those brain wheels spinning, and I want to thank you all very much for your attention today. If you have any questions about this lecture, you know where to find me at laurar@nipissingU.ca, and take care until next time. Bye for now.